Welcome to the Neurology Live Mind Moments podcast. Tune in to hear leaders in neurology sound off on topics that impact your clinical practice. I'm your host, Marco Meglio, editor with Neurology Live. In this episode, we sat down with Dr. Jonathan Parker, an epilepsy and functional neurosurgeon at Mayo Clinic Arizona, to discuss the promise behind stem cell therapy in epilepsy. Parker, who also serves as an assistant professor of neurosurgery and neuroscience, spoke about the origins behind this approach, the different types of cell therapies in development, and the challenges the clinical and research community still faces. In addition, he provided context on a first-in-human study of a cell therapy he is currently helping lead, as well as some of the more unknown and lesser talked about positives from cell therapy approaches. Let's dive in.
Well, I think it's a very exciting approach because it has the potential to be a regenerative therapy to treat our patients with epilepsy. So many of the therapies that we have in our surgical toolbox right now, including open temporal lobectomy or laser ablation, can be very safe and effective, but they are destructive, irreversible procedures, and they can come with some cognitive side effects for a subset of patients. And we get into a scenario where we know we can help a patient's seizures, but there are risks to cognition and memory. So all of us in neurosurgery, in neurology, epileptologists, we want to look to the future to see, can we do a therapy that actually can regenerate some of the abnormal neural circuitry, which is dysfunctional in epilepsy, and provide our patients dramatic seizure reductions or hopefully seizure freedom, and get them to the point where they don't have to have a destructive procedure. I think it's very exciting to look back to the sort of basic science of why we're interested in cell therapy at all in epilepsy. And I think it comes down to the fact that what we've learned over time, and this is not my work, but work of many, if not hundreds of basic scientists and translational scientists over the last couple decades, is understanding this concept of the role of different neuronal subtypes and populations in epilepsy. And I think at a high level, we've come to understand that there's an imbalance between excitatory and inhibitory neurons. And I would say over the last decade, especially in mesial temporal lobe epilepsy, obviously one of the most common forms of adult epilepsy that we see, there's this imbalance of excitatory and inhibitory neurons. And what we've noticed, especially in a subset of patients that have mesial temporal sclerosis, or an imaging finding of a sclerotic, globular, abnormal appearing hippocampus, there's actually loss of some of the inhibitory neurons, so-called interneurons, which make GABA neurotransmitter that tends to be inhibitory in most contexts. And we think that in the pathophysiology, that interneuron loss is important. So the hypothesis stood, well, if there's loss of interneurons, and that's associated with the development of epilepsy, can we repair, regenerate, or put in new interneurons to restore that abnormal function, restore that excitatory inhibitory balance to hopefully stop seizures or dramatically reduce them? So I think that's the preclinical rationale, and that's a lot of science that's come out of basic science labs and both small animal work and larger animal work, and now being translated into a first-in-human study for intrahippocampal transplantation of human interneurons.
Early experience with cell transplantation has primarily come in the Parkinson disease field, although there is a lot of emerging research in stroke and epilepsy as well. Over time, researchers have gained a better understanding of the pathophysiology of epilepsy, including which specific neuronal cell types are abnormal or lost in the brain. Here's Dr. Parker on the emerging research of cell therapies in epilepsy and the optimism behind this approach.
Well, to date, the biggest experience we've had for human cell transplantation for neurologic disorders has really been Parkinson's disease and stroke, and several of those trials have occurred over the last decade plus. There was initially a lot of excitement in some of the human fetal dopaminergic cell transplant for Parkinson's disease, but there was obviously limitations and side effects and other unintended consequences that came with some of those early fundamental trials. It's probably too soon to sell with stem cell therapy for stroke, The science is still evolving there. What are the neuronal cell types that are lost? Is it the primary motor neurons? Is it the supportive cells in the microenvironment, the astrocytes that are in the gliotic scar? And there's some innovative work being done at Stanford University and Gary Steinberg's group in that domain and others. So I think that's a very exciting area to move forward. What I would say, though, how the story is a little bit different for what's happening now in epilepsy is I think that the field was very methodical in how they looked at the pathophysiology, very well characterized in both small animals and in humans what cellular constituents were abnormal and or lost, and then have taken a very directed approach at saying, this is the very specific cell type that we think is dysfunctional or lost. We're going to engineer embryonic stem cells to turn into a terminal fate to very specifically, in a pure fashion, be that type of neuronal cell. And the folks that have been working on this have really done an extended amount of due diligence in the preclinical work to genetically characterize these cells at the transcriptomic level, at the migrational level. Where are they going? How far are they going? So I think the paradigm shift that I've seen in independently looking from the outside into the development work for stem cells for epilepsy is that there's been an even closer laser focus on the exact cell type that we need to get in there. And I think a lot of that's because the techniques and technology of cell preparation has advanced, probably advanced tremendously from those initial fetal stem cell trials for Parkinson's disease, for example. And some have suggested that those may have been a more heterogeneous cell population that was not as terminally differentiated. That may have been a limitation there and potentially led to some of the off-target effects. Whereas in the epilepsy program, the dominant epilepsy program that's out there that started the first in human trial, I think that it really makes a lot of sense because there's a focus on a pure cell population to address that pathophysiologic mechanism which has been well characterized. I think for stroke, something like that is necessary, but in my read of the science, I think it's still evolving. I think there's very rigorous science being done. But stroke is complex, right? We have subcortical and cortical stroke. We have different volumes of stroke. We have differences in the penumbra around the stroke and what's the core, what is a moderately injured zone, what is a still functional zone. And then there's the time domain dynamics of hypoperfusion. Are stroke patients chronically still hypoperfused? How to run their blood pressure? All of these questions which we modify the effect of a stem cell therapy. In my view, I think the stroke stem cell therapies are a little bit more complicated. I'm very optimistic, and obviously it's a very deserving population. And from a public health perspective, something that we definitely should focus a lot of resources on. But for epilepsy, I'm terribly optimistic that this will make a big dent for our patients.
The NTE 1001 study is a first-in-human trial of a stem cell-based treatment specifically targeting human interneurons. The drug candidate NRTX-1001 originates from preclinical research on interneuron loss and is being advanced by the company Neurona. Parker, who leads one of the trial sites for the agent's phase 1-2 study, discussed the outline of the trial and the innovation of this investigational agent.
So obviously this is a first-in-human stem cell trial. The cell product is the genesis of many investigators' preclinical science work that has looked at human interneurons and is now being carried forward by a company. So that's Neurona. So this is an industry-sponsored trial, and there's many sites across the country. And it's taken that knowledge from the preclinical science about interneuron loss in Neurona, in the preclinical colleagues that did the work that led into the formation of that company, have spent a lot of time trying to understand the transcriptomics, the nature of those cells, the nature of how those cells change over time when they're injected into both small animal and large animal models. So the idea is that these are human embryonic stem cells that are then sent down a developmental fate so that they have turned into an interneuron subtype that is very well characterized at the transcriptomic level, with the idea that these cells can then not go backwards, right? The feared complication that all of us, worry about is, does it go backwards? And resume pluripotency? Does it turn into a teratoma? Does it turn into a different neuronal subtype that has an unintended effect? God forbid, an excitatory neuronal subtype. In my read of the preclinical data, I've seen no evidence of that. So I think that's very exciting. And looking from the outside in what's gone into preparing this cell type, it makes a lot of sense to me that this is a strategy that makes sense. Because if we don't have those assurances from the preclinical data, that these are cells that cannot in fact go backwards, then that's obviously concerning. The structure of this clinical trial, there's interval results now that have been released obviously in the public domain. But to summarize in short order, there's been a focus on patients that have mesial temporal sclerosis, like we talked about earlier, that globular, sclerotic, abnormal appearing hippocampus that oftentimes is smaller in volume and may have additional T2 flare signal there that we see in a significant subset of our mesial temporal lobe epilepsy patients. And the clinical trial involves making a very small incision on the back of the head, a very small burr hole or opening through the skull, and then using precise stereotactic techniques to deliver a cell delivery cannula to the hippocampus, and then a very slow, methodical, controlled set of cellular deposits so that we deliver those cells into the hippocampus. As it stands now, this is a cell therapy that is from another human being, is from a human embryonic stem cell line. So it does require immunosuppression. We don't know if these patients will need long-term immunosuppression. There's some sort of early signal that they may not, in fact, need that. So I think it's tremendously optimistic for the future that autologous stem cell therapies such as Aspen Neuroscience and others are working on that for Parkinson's disease, where the stem cells are actually made from a person's own germline or from other hematopoietic stem cells or adipocyte-directed stem cells that have then been taken into a neuronal lineage. I think that's a very exciting approach because then it sort of takes away the immunosuppression part of things. So I think that there's a lot of innovation and science being done to make this out of the patient's own cells, and I think that's probably the future.
We're going to take a quick break from our conversation with Dr. Parker to keep you up to date on the latest developments in neurology. Welcome to the Neurology News Minute. Here's what you may have missed since our last episode. First, on November 26th, Anavex announced it submitted a marketing authorization application to the EU for blarcamesine, a small molecular activator of the sigma-1 receptor, as a treatment for Alzheimer's disease. The application was based on data from a phase 2-3 placebo-controlled 48-week study of 508 individuals with a confirmed diagnosis of early-stage AD. About a week later, on December 3rd, the Relieve CM2 study was initiated. The study, an FDA-approved investigational device exemption pivotal trial, tests the therapeutic effects of Shiratronics neuromodulation device in patients with chronic migraine. And lastly, on December 4th, the FDA granted breakthrough therapy designation to Stoke Therapeutics' investigational agent STK-001 for the treatment of genetically confirmed Dravet syndrome. The company remains in discussions with the agency for plans on a phase 3 registrational study, which are expected to be released by the end of this year. For more information about these stories, head to neurologylive.com, or you can check out the corresponding link in the show notes. And now, back to our conversation with Dr. Parker. There are currently no approved cell therapies to treat epilepsy, as there are still several unanswered questions and uncertainties behind their use. Here's Parker on the challenges and roadblocks in advancing stem cell therapies for epilepsy, as well as the need to balance ideal and realistic clinical trials. He also spoke on the role of patient advocacy and community engagement to reimagine and refine what trials for stem cell therapies should look like.
In terms of roadblocks, there's so many unanswered questions, right? How many cells? Where to put the cells? What is the right dose? Would patients benefit from redosing, right? We can think of a multitude of different questions that are likely going to be relevant in the future to optimizing the therapy. But first and foremost, we have to focus on safety, right? We have to ensure that the cell product is safe And that then obviously the next step is proving efficacy, and that's how our clinical trial system is designed. I think the challenge, though, is for these novel cell therapies, especially something that requires a surgery, there's an ebb and flow between regulatory agencies, what the FDA sees as an ideal clinical trial design, what clinicians see balances beneficence to the patient and do no harm versus risk to the patient. And then there's most importantly, the patient factors. What do the patients need? The patients need a therapy that reduces their seizures or gets them to seizure freedom. This clinical trial, and clinical trials no doubt that will come down the line, is geared towards patients that are having seizures despite the best medications that we have to offer. And they're desperate for a new therapy. But when you look at the regulatory side, there's a push for clinical trial designs that have sham surgery controls in things that may make sense if we're thinking conceptually and scientifically. And we all know about bias effects and expectation effects and placebo effects, and those are real, and we have to consider those. But we have to achieve a balance or homeostasis between what is a perfect, ideal clinical trial design, understanding true efficacy and true safety, and also what the patients need. A sham surgery trial to a patient that has medication refractory epilepsy, where they have to undergo a period of not getting therapy, is a very challenging thing to talk about. It's a very challenging thing from a patient perspective and a family perspective to engage in. So I think it requires a continued dialogue from our communities, the neurology community, the neurosurgery community, the regulatory community, the FDA, about for these novel cell therapies that are going into the central nervous system, is sham surgery necessary in the most appropriate control, or are there more innovative clinical trial designs that allow us to be more nimble, move these therapies along more quickly, but also ensure sound science and sound safety? I think that's the biggest roadblock that we as a community have to challenge, or we as a community have to address. But I'm optimistic. There's a lot of energy. Patients need the therapy. The patient advocacy groups, Epilepsy Foundation, others are very strongly involved in this, and I would encourage them to continue to be so. And I think it will require an ongoing dialogue between the patients, the clinicians, and the regulatory agencies to reimagine what these clinical trials would look like going forward.
There can often be misconception or skepticism about a new novel treatment approach when it is introduced to the clinical community. In the discussion, Parker spoke on the importance of integrating stem cell therapies into the broader treatment framework for epilepsy and similar conditions. He stressed that these therapies will not replace traditional medications, but will complement them. Here's Dr. Parker on some of the potential gaps in understanding or misconceptions about stem cell therapies for epilepsy within the clinical community.
I think one perspective that's going to be important is that cell therapies are part of the toolbox, right? And we're sort of anxiously awaiting, especially for epilepsy, for stroke, for Parkinson's disease, for that first FDA-approved cell product, right, that we can deliver to our patients. And I'm very optimistic that that will happen soon. But we know with any new therapy, we have to understand how to apply it, patient selection, what is the combinatorial role of when to deliver the therapy and how to manage medications. So even though cell therapy in some ways will be driven from a delivery perspective by the neurosurgeons, it means we definitely need to continue to work together. I don't see it as where the patient goes to the neurosurgeon, they get their cell therapy, and the neurosurgeon is no longer involved. This is a crosstalk between neurology and neurosurgery and other subspecialties that touch on the neurosciences. I think there's many unanswered questions about how chronic medication management in the epilepsy population should be done with stem cell therapy. Same thing probably for stroke rehab and also for Parkinson's disease. So I think that the continued tight collaborations between neurology and neurosurgery is absolutely essential. And I don't see cell therapy as necessarily a one-time treatment and then we ignore medications. I think medications will continue to play a role and potentially we'll understand better as time evolves what is the synergy between medications and cell therapy. How do we maximize clinical effect, minimize side effects, right, and get our patients restored to their quality of life? And we all know sometimes medications are a strong part of that equation, that they have clinical benefit, but oftentimes, especially in epilepsy, high side effect profiles, right, which add to the morbidity of the condition. So I think that interplay is something that we need to continue to work on together. So that would be, I think, just the framework that I look as the cell therapies. It's still very much multidisciplinary between our specialties.
Wrapping up our discussion with Dr. Parker, we continue to talk about the potential incorporation of cell therapies for epilepsy in the future. Here's Parker one last time on the unique nature of testing cell therapies in children, the desire for restorative therapies, and the promise of early intervention.
So I think epilepsy is a very unique condition in that it oftentimes will start in younger ages, in children or in young adults. So I think when people look at what therapies do I want to engage with, what do I want to do, it may be a different mindset than Parkinson's disease or stroke, where it quantitatively happens in older patients, right? In epilepsy, people may be looking at 80 years, 90 years of life expectancy with this disease, right, with medication management alone. So I think the potential to have something to change that trajectory early on in life, a restorative or regenerative therapy, is very exciting to our patients. To think about that it may not be medications for the rest of their life, or medications may play a smaller role in the management of that condition. I think that plays into both people's interest in being involved in the clinical studies and clinical trials, but also in their interest in engaging with a therapy like that, which in our current iteration, it still requires surgery, right? And there's innovative work looking at different ways to deliver cells, blood-brain barrier disruption, endovascular approaches, other more minimally invasive approaches through the CSF. And I don't think we're quite there yet, and we think probably for epilepsy, you have to be closer to the source of the initiation of the seizure and the ictogenesis and in the hippocampus, at least for mesial temporal lobe epilepsy. But I think a lot of patients are looking for something regenerative or restorative that will change the trajectory of that condition for the rest of their life.
Thanks for listening to this episode of the Neurology Live Mind Moments podcast. To support the show, be sure to rate, review, and subscribe wherever you listen to podcasts. And as always, for more neurology news and expert-driven content, visit neurologylive.com.